And so again, thank you to the IAAM organizers, and again for the opportunity to be here, for the invitation. And so we'll talk about these nanoprobes and different challenges approaching it through solution phase modification of these different types of nanoparticles. And so I'll talk about some of the challenges, and then I'll go into one of the approaches that we're taking for modifying those. And so we've seen an array of materials by design, such as in the previous lecture, but also in the sense of nanomaterials. And so there's really too much to review. And so I'm just going to pick out a few things to highlight. And so you take the case of quantum materials, quantum dots in particular, a lot of different what I call solution phase techniques in order to prepare these. And so you can come up with an array of different optical properties, and these have been pursued now for quite a number of years. The surface chemistry, though, is the thing of interest for today. And so how to modify these. And there's obviously, halide perovskites and other materials now that are also quite popular in the literature. And another area is looking at upconverting particles. This is an area that we also participated in collaboration with Professor Branda here at SFU. And so just as an example, upconverting particles are also optically interesting. And so depending on the intensity of a near-infrared laser, you can actually get two different emissions coming out. And that was utilized to be able to activate a ring opening effect and ring closing on a molecule that was dispersed in solution, but also then bound to the surface of those particles. And so, again, there's a lot of different functional materials out there that you can look at that offer up a range of properties, and it's desirable to be able to stabilize those or to be able to anchor specific chemistry in proximity to whatever your particle is. And so that's more of what we want to talk about today. So looking at engineered nanomaterials, I want to look at how to harness the surface chemistry, because in order to use the property, you really have to harness the surface chemistry. So in the background, you're going to see some pictures of the surrounding countryside of British Columbia. I love taking photography, and so you'll see a few of those throughout. In surface chemistry, you take a nanoparticles, make it by solution phase. Oftentimes we start with one surface chemistry, but we want to end up with a different chemistry at the end of the day. And often in the reports and literature, it's reported as, I put X molecule in at X concentration and temperature, and therefore I must end up with the final product. It's not always the case. In fact, many times it's not the case. And so is it really that we have this material to start off with? Or do we have some inclusions where we have molecules that are not quite adhered properly or are facing the wrong way? Or we have inclusions of some of the original molecules? Or is it even worse than that, where we have many more defects? And so, how do we measure this? And how do we control for this? And so this is an area that I'm interested in, is looking at the quantification of the functional groups that you have on the surface of a particle, but also how do you assess a population of particles? And so versus individual particles, or versus just an average overall. And so there's excellent work being done on a variety of targets herein. Here's another consideration. And so a matter of scale. So I draw these cartoons, and sometimes we draw fancier cartoons with even more functionality. But if you take the consideration of what is the length of a molecule, a typical molecule that you might put on the surface of a nanoparticle, if it were on the order of a nanometer, and this is then a 10 nanometer particle, it shows you the scale at which you're at for a 10 nanometer particle. If you take that up to the upper range of nanoparticles, and you say, let's look at a 100 nanometer particle, now it's obviously the particle's off the screen in the proportion of scale. And so it shows you really how much demand there is on the properties that we're pulling out of that molecule on the surface of the nanoparticle. And so a lot of it comes down to that red group on the end. We want to functionalize the nanoparticle with that new functionality. And so a lot of the properties are going to depend on how many of those and the quality of the packing that we have here and so forth. But there are further complications. And so one of those complications is that depending on the material, we're either dealing with a semicrystalline or amorphous surface, or we might be dealing with crystalline facets. And so when dealing with faceting, then each of the different facets can have a unique packing of molecules. And I'm not going to go into the details therein, but also the edge sites can also present then another type of packing or another type of defect. And so really the surfaces of nanomaterials can be quite diverse when you get down to the atomic scale. One way of probing that, this is a technique we demonstrated before, and the particles that we're using in the background are, we start off with a CTAB capped or a surfactant capped nanorod, gold nanorod. We put an intermediate polymer on there, and then exchange that off for a thiolated, single-stranded DNA. And so the sequence is down here below. And so that we draw this ideal structure, and it looks like the DNA is fully covered the nanorod. But we know that that's not always the case. And so we used a gel electrophoresis, and these are bands run in gel electrophoresis where we load the samples of the gold nanorod solutions here in the top. Each of these wells is a different solution. And we load this with a high level of salt in the initial well, except for in the first one. And so in the first column here, we have no salts added. The other ones have a high concentration of sodium chloride and magnesium chloride added in. And the experiment is we start off with, we've got the polymer coated ones, and then we've got various degrees of where we've created a ratio of thiolated DNA to the gold rods, and doing that exchange from the polymer to the thiolated DNA. And so what we find is that depending on the ratio that we have, and we're well in excess of thiolated DNA. What we find is that there's actually there's this aggregation here, and then the red that you see, this is from the plasmon of the gold rods. And what we find is that there's more and more smearing, but as we get up to higher concentrations, then we get a banding that's very similar to what we started off with for the polymer coated rods that were dispersed without salt. And so the salt is leading to some aggregation there when we have an unstabilized surface. But when we are not uniformly coated with the thiolated DNA, then we also see a distribution in how the rods move through the gel. And so it's, there's still some rods that are left in the other parts of the gel. So is it perfect? No, it's not. And so, but at least you see that now this is a visualization of really what's happening to the population. And that's really of interest. And so I think there's a lot more just simple techniques like this that can be developed for probing really the population and what's happening to the population of different nanomaterials. And obviously the gel electrophoresis doesn't apply in all cases. So we want to take a look at extending this to a different class of chemistries, and rather than thiols. I really want to look at oxides. And so one approach has been the use of silane chemistry, and we've used a lot of silane chemistry ourselves in the past. And one of the challenges is that you can end up with multi-layers, as shown here. You can end up with these branch type structures on the surface. And as shown in the top images, a modification of a wafer can lead to very high topography. And so depending on the molecule and how you're depositing the molecule. And so we've used monoreactive and multi-reactive silanes in an attempt to improve the quality of these. But in recent years, we've taken a deviation away from these type of silane-based reagents, and instead looked at how to modify the surfaces of different silicon oxides, whether it's an oxide on a polysilicon wafer or an oxide here on that is a mixed oxide in the scintillation vials, and to modify them using alcohol-based reagents. And the idea is that the alcohol and the silanol on the surface of whatever surface we're dealing with, and we're going to focus on silicon-based oxides today. We undergo a mild thermal treatment with water as a byproduct, and then ideally end up with such a coating like this. And so let me get So in the two vials that you see here, these are scintillation vials coated by this process. The one on the left is not coated, the one on the right has been coated, and you have a glycol solution, and you see that it de-wets quite well for the one on the right. And so, you know, in the simplest form, this is a way of treating your either glass slides or scintillation vials. All right, and so we've extended that off to look at a range of different surface chemistries. I'm not going to go into that in detail, but we have looked at a range of different surface modifications, and further work can be followed here. We follow that by a range of spectroscopies, whether it's looking at infrared spectroscopies and XPS, as well as other techniques. We also have looked at mixed layers, and so herein, you know, a ratio between a fluorinated alcohol and a hydrocarbon based alcohol, and looking at water, toluene, and hexadecane contact angles, how they change as a function of changing these the ratio of these compositions. And so the idea is going from something that is 100% of one to 100% of the other, so a hydrocarbon to a fluorocarbon. And what we find is that you know, changing this out, we see a gradation in the contact angles that we get for the toluene and hexadecane, and not surprisingly, the water is more flat as far as its response. And so, it just shows you that, you know, this is a simple way to be able to tune the composition by simply mixing the ratio of these species in the original solution, and then these layers that we make, right, are formed by a thermal process. We can do that in an oil bath or a microwave or other processes. We've extended this off to indium tin oxide surfaces, and grafting on a range of different molecules on there to change the work function. And just briefly, we used one test of this we used as a working electrode in an electrochemical probe, immersed into an electrochemical probe, and then looked at the cyclic voltammogram, or the response to potential cycling, up to 10,000 scans. Well, the original ITO is fairly consistent, let's say. And if you modify the surface, then that modified here, this is one of those alcohol modified ones, versus you know, one that is coated with a phosphonic acid, or one that is coated with a silane. And so you see some destabilization in those cases, but fairly consistent in the case of the alcohol modified one. There are other changes, as you can tell, in regards to the intensity relative to the pure indium tin oxide, and that's not surprising. There is a change in the resistance at that interface. Why is this interesting? Looking at for example, ITO interfaces are found in a lot of different devices. We see them in phones, and we see a hue change over time. And so we also see it in displays, this vignetting that can happen at the edges of displays. And so the idea is to avoid that. And there's many reasons why that comes about, but you have some adverse chemistry on the surface. Looking at the XPS change over time after those 10,000 cycles, but also looking at a change in color, the silane-based coating ends up with a blue hue. You get an oxide depositing on there, and you change your dielectric properties. Versus that of the alcohol modified one, we largely retain the same reflective properties of the indium tin oxide, and the XPS analysis is fairly consistent as well. There is a change between the pure indium oxide that is cleaned. You still have a little bit of residual carbon on that. There is a carbon layer that is consistent on these modified electrodes. So moving into nanoparticles, then, taking a look at these alcohol-based surface modifications, we're first going to look at magnetic particles. And magnetic particles have been pursued for a range of different applications. One is MRI contrast agents. Another one is looking at what's called thermal ablation, the use of an applied AC magnetic field to then trigger the release of heat locally from the nanoparticle. And there's a couple different mechanisms of release of heat. And so that heat can then be used to trigger reactions locally, or phase changes, a change in solubility of a species. And so this has been an area of increasing interest for magnetic nanoparticles. So for us, we're interested in the surface chemistry, as I said, and so hadn't mentioned it before, but the students that are leading each of the projects, they're the ones in the picture up in the right-hand corner, and their citations are in the bottom left. This is one where a previous master's student worked on modifying the surface of a magnetic particle. We put on a thin silica coat through a sol gel process, and then modify the surface of that using the alcohol-based reagents. And so the alcohol-based reagents, I put some of the chemistry that we've been using down below. And so fairly low temperature. We've done some even lower temperature reactions as well. And we let it run for a fairly long reaction time. That is a limitation of this technique. But this was one technique where we used a longer time and an oil bath. We've since come back and done this with microwave processes as well, and we can speed this up quite a bit. How do you know how much of the thiol is on the surface? One way, this is a previous report in the literature, looking at polystyrene that has a thiol functionality. They used the Ellman's test and showed that this reagent, when reacted with the free thiol and the polystyrene, could then release this reporter into solution. So this TNB actually has a change in absorbance property, and you can then monitor its release and quantify then the formation of the disulfide on the surface of the polystyrene. And we extended this off to look at our magnetic particles and the modification specifically of the thiols therein. And taking a look at this, how do you estimate how many functional groups you have based on literature reports, looking at from the sol gel process. And this does not consider the porosity of these layers, and so that'll have an implication in the quantification. But as a one estimate, it could again be an underestimate. We have 4.6 silanols per nanometer squared. And so, a cartoon version of that would look something like this. And so again, not drawn to scale. And modifying that with these alcohol functional groups, condensing those onto the surface, we end up with, for the thiols, using the Ellman's assay to quantify, we end up with about 0.5 thiols per nanometer squared. And so it shows you that, yes, there are some reactive groups that didn't react. In this particular case, it's about 12%. We've since been able to come back and then tune this, and we can actually, this is one of the nice things about this approach, is that although you're not getting 100% coating, and I think there's multiple reasons why that we're not, but you can tune exactly what the loading is, and that I think is a key component. If you look at the control example where you incubate it with the thiol at room temperature, we still go through the same purification process. Then we end up with one of those thiols per the same area. Probing that further, we take these core shell particles, and then we combine them with gold particles, and each are soluble or dispersed in opposite solutions. One is we've got these iron particles in an IPA solution, and the gold is in an aqueous solution. If you combine the two together, they will crash out the aggregate in solution. And so this is the key thing, was to be able to stabilize this process. And this is another area that I'm interested in, is looking at the assembly of things that are otherwise unstable, and trying to make something that is, at the end of the day, stable and colloidally stable is what I'm implying. And so we were able to make these gold decorated particles. And to show you what those look like, turn off that laser pointer for you and take a look at this. This is a tomography image. We've done multiple tomography images looking at then the gold, and it is coating on multiple surfaces. We're not getting one gold per thiol because of electrostatic repulsion between the gold particles, but and there's many more thiols on the surface than that. But we are able to cover over a range of the surfaces, and we can tune that loading. We've shown that you can come back and tune what that loading is. But that's not at all of interest. I wanted to show where we can go with the diversity of the chemistry. And so, you know, I want to extend this to a different type of nanomaterial. And in particular, I wanted to look at lithium niobate. And so we're interested in the formation of single crystalline lithium niobate and the modification of the surfaces therein. And we've developed a few different synthetic routes where we can look at the tuning of the size. This is one through an Ostwald ripening process. Of interest, for lithium niobate nanocrystals, they can be used as probes, not just in biological systems, but in other systems where you use near-infrared light. And this is using a femtosecond pulse laser, and we can get out visible light. And this is tunable. And so this second harmonic process can be applied to these nanoprobes. Now, we've shown that there's a number of different synthetic routes and a number of different impacts of precursors on making these lithium niobate particles. To read this diagram, we have solubility issues with some precursors here. The purity of the lithium niobate is the highest over here in this region, and it's also where we find that we don't have these impurity phases either. And so really it comes down to these precursors here, the ACAC, the ethoxide and the hydroxide. These are some of the best reagents. And so this is a key thing is you need to have uniform, and you have size control and control over crystallinity. And so as one example, extending that off, looking at the hydroxide precursor, starting with the ACAC, we form the hydroxide precursor. In this case, we're able to form some very small particles. I'll characterize those further on the next slide. And we're able to see that we have a tunable second harmonic response across the visible range. And so another aspect of this is looking at 30 nanometer particles as just one example. And these particles, although a little bit irregular in the surface, we found that we're able to make some single crystalline materials. And so we've since tuned that synthesis and been able to make them a little bit more cubic in size. But we wanted to diversify the surface chemistry. And so second harmonic particles are fine, but you have to have a second harmonic microscope, a second harmonic monitoring microscope, meaning a femtosecond pulse laser and so forth, appropriate detector, in order to track these. So we want to put a label on there to track them using regular fluorescence. And so then you can look at the co-localization of this to other things of interest. And we want to be able to obviously stabilize these for physiological or other adverse conditions. And then we might want to add in targeting or delivery. And so how do we diversify the surface chemistry? And so one approach that we chose was to look at a thin silica layer and then using the alcohol condensation reaction. And we'll talk about that more in the next slide. Without that coating, the lithium niobate can undergo leaching of the lithium out of there or aggregation of the particles. And of course, they're not labeled for tracking. So the modification that we did was looking at putting first a carboxylic acid modified alcohol onto the surface and having this carboxylic acid functional group that would be available for further reactions. And that's a key thing. There is some speculation in the literature that this functional group might actually react the other way with the surfaces, but that's not what we've seen based on the spectroscopies that we've done. And that's interesting. And so it opens up the avenue that, with the alcohol condensation, there's a range of functionality that you can put on the surface. And so this is a demonstration paper to really show that diversity of where you can go, but there's much more chemistry that we've done on the surface. So this carboxylic acid group can be used with EDC coupling to put on a PEG with other functionalities, or to put on a dye. This is a dye, a proprietary commercial dye that then has a tunable not tunable, but a red emission. And so you see that here, whereby without the dye on the surface versus with the dye on the surface, we're able to tag these particles. This is a colloidal suspension of the particles. There is a modification of the lifetime for the dye that is attached to the particle versus that is free. And so versus also we have a control in here where we have the free dye, but also the dye without doing the EDC coupling, just freely suspended in solution with these particles. And so when coupling to the surface, it's not a surprise that we've modified the lifetime. And so we've done a range of spectroscopies to probe that as well. I'm not going to go into that. The thin silica coat and the process of modifying the surface chemistry could lead to adverse changes in the properties, but we're able to show that the second harmonic property, the tunability of looking at the second harmonic, looking at that tuned emission, or scattering, I should say, is preserved for the second harmonic particles. And so the lithium niobate is not adversely affected. And we've seen that in the Raman spectroscopy as well, that we're not leaching out lithium. And so the ratio of lithium and niobium are consistent. And so with that, what's next? I think there's a lot of other surface chemistries to pursue with these. And with that, we really want to acknowledge the work of Faryad and Melissa, both of who've been working on extending this, and we've demonstrated that we can really tune the loading of those thiles. This is our group photo during the pandemic. We haven't been able to get together as a team other than virtually, so it's great to work with a lot of really talented young individuals. And so just give acknowledgement to all of them here. I wasn't able to talk about nearly all of their work, but was able to introduce just a little bit of it to you. We work with a lot of other talented teams around the world, and that's really our pleasure is to work with other individuals and to augment their chemistry with our own solutions. And with that, really want to acknowledge the land on which we work and the different territories on whose land SFU resides. And also, again, want to thank IAAM for the invitation and opportunity. And thank you for listening.